Hello and welcome everybody to my channel Lumini Bioentrance, and today we will be discussing West Bengal SET Life Science question paper. This is the 25th SET exam and I have solved this paper with explanation. This is part 1 of the explanation. If you are new to my channel, please do like, share and subscribe. Coming to the first question. Which one of the following has biradial symmetrical body? They have asked biradial, not bilateral. What is bilateral? Bilateral is to divide an organism into two equal parts. Biradial symmetry occurs when an animal can be divided into four parts about a central axis, and each opposing segment, that is segments which are opposite to each other after division, are identical to each other, whereas segments that are adjacent to each other after division are dissimilar. That is biradial symmetry. So planaria show bilateral symmetry, fasciola show bilateral symmetry, branchiones show bilateral symmetry, whereas metridium it shows biradial symmetry. So the answer here is option 4. The next one. The membrane phospholipids form bilayer when water is available on all sides due to its amphipathic nature. Amphipathic means they have hydrophilic or polar ends or water-loving ends and a hydrophobic end. So the most abundant membrane lipids are the phospholipids, and phospholipids spontaneously form bilayers in aqueous solution because of their amphipathic nature. The hydrophobic tails will be on the interior of the membrane and the polar head groups exposed on to water. So answer is option A. Which of the following is not a step in southern blotting technique? Separation of DNA fragments on a gel. Yes. Ligation of DNA to a vector. Southern blotting is not a cloning procedure. So you need not have to go for any ligation. It is a method to detect specific DNA molecules from many other DNA molecules. So that is ligation. So answer is option B. Then next one. The population density of an insect species increased from 50 to 60 in 1 month. If the birth rate for the period is 0.4, then the death rate is. So N0, that is initial, it is 50, and NT, that is final, is 60. Birth rate is 0.4. What is the death rate? So you have to calculate the growth rate first. NT minus N0 by N0, you will get it as 0.2. Growth rate is equal to birth rate minus death rate. So growth substitute. Growth rate is 0.2, birth rate is 0.4 minus death rate. So death rate is 0.4 minus 0.2. So 0.2 is the answer. Answer is option A. Then next one. A major goal of Golden Rice Project is to supply consumers in rice-based societies with recommended daily intake of. So golden rice, it is a rice produced through genetic engineering to biosynthesize beta carotene, which is a precursor of vitamin A, in the edible part of the rice. It was intended to produce fortified food to be grown and consumed in areas where there is shortage of dietary vitamin A. Answer is option C. Then next one. For induction of somatic embryogenesis in higher plants, the cultured friable callus tissue need to be exposed to. So somatic embryogenesis is the development of embryos from somatic cells, that is from plant cells which are not germline cells. From normal plant cells but not germline cells. So for that induction of somatic embryogenesis in higher plants, the cultured friable callus tissue need to be exposed to high concentration of auxin for a short duration. Answer is option A. Then next one. Glucose 6-phosphate is a substrate or a product in all of the following pathways except glycolysis, gluconeogenesis, beta oxidation. Beta oxidation is for fatty acids, not for glucose. So the answer is option C. Which of the following statement is correct? Numerical taxonomy aims at determining phenetic relationships between organisms and taxa. That's correct. APG system of classification, that is angiosperm phylogeny group system of classification, is established by taking information from recent molecular studies. That is also correct. Phylogenetic classification is constructed in such a way that all the descendants of a common ancestor should be placed in the same group. That is, group should be monophyletic. That is also correct. So, answer here is option D. That is 1, 2 and 3 are correct. Then next one. Match the non-protein coding RNA molecules present in genome with their function. Non-protein coding RNA. snRNA, mRNA splicing, snORNA, pre-rRNA splicing, XIST, that is in chromosome inactivation, X chromosome inactivation, miRNA, regulation of gene expression, and rRNA, protein synthesis. So the answer here is option C. Then next one. Which one of the following complexes of electron transport system does not account for the pumping of protons from the mitochondrial matrix? It is complex 2. This is because complex 2 directly passes electron to coenzyme Q rather than using it to pump protons. Then, chemical that is required for maintaining the protoplast in intact condition during protoplast culture is mannitol. Answer is option B. Next one. Individuals with greater mass have smaller surface area to volume ratio, which helps to conserve heat. This is known as, so larger animals, they have lower surface area to volume ratio, so they can radiate less body heat per unit mass and stay warmer in cold climates. This is in the case of polar bear. Now what does Bergman rule tell? Bergman rule tell that organisms with larger sizes are found in colder altitudes, while those with smaller sizes are found in the warmer region. Now in cold climates, they have larger surface area to volume ratio, so they radiate less amount of heat and they can stay warmer in the cold climates. So individuals with greater mass have smaller surface area to volume ratio, which help to conserve heat, and this rule is known as Bergman's rule. Answer is option B. Then next one. In a cell fusion experiment, a cell in S phase was fused with cell in the G1 phase, G2 phase, M phase. The fused cells were grown in medium containing tritium labeled thymidine. Maximum radio label was obtained in the S phase cell fused with. So when a cell in S phase is fused with cell in G1 phase, what happens is the G1 phase is stimulated to synthesize DNA. This is because cytoplasm of S phase cell contain factors that initiated DNA synthesis in the G1 nucleus. It cannot be G2 because in G2 phase DNA synthesis has already taken place. In M phase also it is the same way. So the answer is G1 phase cells. Then next one. In antibody molecules, variable regions are present in. So this is the heavy chain, the blue one, and the green one is the light chain. So this is the variable region of the light chain. This is variable region of heavy chain. These are the constant regions of the heavy chain. So in antibody molecule, variable regions are present in both heavy and light chains. Answer is option C. Then which of the following is a constitutive promoter for expression of transgene in monocotyledonous plants? So strong constitutive promoters, they can deliver high level of expression of the transgene in almost all the tissues, including the developmental stages in a plant. So, which of the following is a constitutive promoter for expression of transgene in monocotyledonous plants? Is 35 as promoter can be used. Ubiquitin gene promoter can also be used. They both are strong constitutive promoters. So, answer is A and B. Then next one. The major detoxification reactions in the phase 1 involved all of the following except. So phase 1 reactions of drug detoxification occurs by oxidation, reduction, hydrolysis, cyclization, decyclization, addition of oxygen or removal of hydrogen carried out by mixed function oxidases often in the liver. So oxidation is there, acetylation is not there. Answer is option B. Then which of the following diseases is associated with protein misfolding? Cystic fibrosis is associated with protein misfolding. Transmissible spongiform encephalopathy, prions, again misfolding. Gaucher's disease, again misfolding. Phenylketonuria is inherited inability to metabolize phenylalanine. It is not associated with protein folding. So A, B, D are correct. Then next one. Which one of the following statement is not true for the retinal protein bacteriorhodopsin? It is the major photosynthetic pigment of the bacterium Rhodopseudomonas palustris. That itself is not a correct statement. Answer is option A. The pygmy hog and the hangul are the representatives of the following Indian states, that is Assam and Jammu and Kashmir. Answer is option B. Which of the following statement is not true about brown fat tissue? So it is abundant in mitochondria, yes. It is specialized in heat generation, yes. It is specialized for storage of fat. It is not specialized for storage of fat. It is adipose tissue which is specialized in storage of fat. So answer is option C. Then in a brackish water shrimp farm, a farmer detected some shrimps with white spots of variable sizes prominently seen on the carapace. It could be caused due to baculovirus infection. Answer is option A. In a diploid organism, segregation of the two alleles after a recombination event between the given gene and the centromere of the chromosome occur at. So it occurs at anaphase 2. C. In a diploid organism, segregation of the two alleles after recombination event between the gene and the centromere of the chromosome occur at anaphase 2. In anaphase 1, the whole chromosome along with the two chromatids are segregated. But in anaphase 2, the chromatids are separated. So the alleles are also separated after recombination. So answer is, you can see here, this is interphase chromosome, these are the homologous chromosome, this is prophase 1, this is meiosis 1 after recombination, these are the daughter nuclei. So prophase 1, there is recombination taking place in prophase 1. These are the daughter nuclei that you get after recombination, where you can see that this has combined with this has undergone recombination with this, and you get 1, 2, 2, 1, 2. That is 2 chromosomes each in the daughter nuclei. Whereas in meiosis 2, the recombinants are separated. See here, the recombinants are separated in meiosis 2. That is next one. Which of the following serves as the source of carbon dioxide for the Calvin cycle in C4 plants? That is malic acid. Answer is option B. Which one of the following neurotransmitters is responsible for awakening from sleep? Serotonin is responsible, dopamine is responsible, acetylcholine is also responsible. GABA is an inhibitor, so it is not the neurotransmitter responsible for awakening from sleep. So answer is option A, B and C. Acquisition for playing piano is an example of. So there are two types of memory. One is declarative memory and the other is non-declarative memories. Declarative memory is memory that you are consciously aware of, and non-declarative memory is unconscious memory that can affect your behavior. Now this non-declarative memory, it is of two types, that is priming and procedural memory. And in procedural memory is for how to do things like playing piano, playing guitar, how to ride a bike. All these come under procedural memory. So acquisition skills for playing piano, it is a non-declarative memory. Answer is option A. Methotrexate, an anti-cancer drug, is an inhibitor of dihydrofolate reductase. And this dihydrofolate reductase catalyzes the conversion of dihydrofolate into tetrahydrofolate, the active form of folic acid. And tetrahydrofolate, it is necessary for the synthesis of nucleotides of both DNA and RNA. So the answer here is option B. Then next one. Which one of the following does not contribute to the productivity in the marine ecosystem? So productivity is the amount of organic matter made by photosynthesizing organisms in an ecosystem. In marine ecosystem, the photosynthesizing organs are phytoplanktons, dinoflagellates, diatoms, algae, etc. Mollusks and crustaceans, they are non-photosynthesizing. So the answer here is option D. Now next one. Which one of the following statements about the nucleolus is false? It is surrounded by a lipid bilayer membrane. It does not have a membrane. So that statement itself is false. Answer is option A. Then, the aggregated mass of sterile cells located inside the base of the capsules of Pellia is called, they are called as elatrophore. Answer is option A. Then all of the following statements are true with respect to T lymphocytes except they arise from the bone marrow and mature in the thymus. Correct. During maturation, T cells acquire a receptor called T cell receptor on the membrane. Yes. The secretory product of T cells include antibodies belonging to different classes. No. The secretory product of B cells include antibodies, not T cells. So answer is option C. Then, in Drosophila species has 3 pair of metacentric chromosome, 1 pair of telocentric chromosome. This species will have telocentric means centromere is terminally situated so that there is only one chromosomal arm. So 1 pair of telocentric chromosome means one chromosomal arm, and metacentric 3 pairs of metacentric. The centromere is present in the middle of the chromosome and divide the chromosome into 2 equal arms in the case of metacentric chromosome. So 3 pairs means totally there will be 6. 3 into 2, 6 arms will be there. And 1 pair of telocentric means one arm only will be there. So totally 7 arms in the polytene chromosome. Answer is option A. Then next one. Out of the following enzymes mentioned below, which one is activated by acid hydrolysis of the proenzyme form? They have asked which one is undergoing acid hydrolysis of the proenzyme. Usually, the proenzyme is activated by protease hydrolysis, but here it is acid hydrolysis. So, acid hydrolysis is for pepsinogen to get converted to pepsine. So, answer is option A. Then next one. Which of the following statement is not correct of the phenomenon of photoblastism? Photoblastism is the mechanism of seed dormancy, and photoblastic seeds require light in order to germinate. The phenomenon of photoblastism was discovered in lettuce seeds. That's correct. Germination of photoblastic lettuce seeds is regulated by the pigment phytochrome. That is also correct. These are all research studies. Phytochrome regulated lettuce seed germination is thought to be mediated by auxin. It is not auxin, it is gibberellin. That is as per the study. Okay. Red light irradiation induces germination of lettuce seeds. That is also correct. So answer is option C. Then next one. A phytohormone which is produced by plants under water deficient condition and plays a key role in the tolerance response of plants to drought is abscisic acid. Answer is option B. A, B, A. Then next one. Which one of the following learning processes belong to associative learning type? So learning can be divided into two general categories, that is associative learning and non-associative learning. So associative learning is classical conditioning and operant conditioning, and non-associative learning is habituation and sensitization. So A and B are non-associative learning, whereas classical conditioning is associative learning. So the answer here is option C. The restriction enzyme EcoR1 recognizes and cleaves at GAATTC. What would be the approximate number of DNA fragments produced by cleaving E. coli genome of size 4.6 into 10 raised to 6 base pair with EcoR1? So GAATTC is the recognition site for the restriction enzyme EcoR1. Probability that G will occupy the first position is 1 by 4 because there are 4 nucleotides. So that is A, T, G and C we have got, and there are and out of these 4, G will occupy the first position. The probability is 1 by 4. Probability that A will occupy the position 2 is 1 by 4. Similarly for others also. Now probability that this cleavage site occurs on the DNA is multiply all the probabilities and you will get 1 by 4096. Then next comes number of fragments produced by cleaving the E. coli genome of size 4.60 into 10 raised to 6 base pair. So divide 4.60 into 10 raised to 6 by 4096, you will get it as 1123. So the answer is option C. Then next one. If you wish to study the region of binding of transcription factor to the promoter DNA, which one of the following techniques will be the most appropriate? So transcription factor is a protein and promoter DNA is a sequence on the DNA. So protein binding to DNA, if you want to study that, which technique will you use? It is DNA footprinting, that is for protein DNA interaction studies. Chromosome working is done is used to clone a gene from its known closest markers. Microarray is to detect expression of thousands of genes at the same time. Immunoprecipitation is a method to isolate specific antigen from a mixture of antigen using antigen-antibody complexing reaction. So the answer here is option B, that is DNA footprinting. Then next one. Which one of the following statements about analysis of variance is not correct? It is commonly used for dividing the variance into components. Yes. It is used for determining whether the differences from the mean are significant, that is larger than expected by chance. Yes. The analysis involves partitioning the variance and comparing the role of various factors, environmental, genetical, etc. That is also correct. It is calculated by adding the series of values and dividing the total by the number of values. That will give your average, not analysis of variance. So option D is the answer. Option D is dealing with the average, not analysis of variance. And next one. If a sample DNA is found to have the base composition as follows: adenine 40, thymine 22, guanine 21 and cytosine 17. Then which of one of the following conclusions will be most appropriate? The given DNA is single-stranded DNA molecule. That itself is correct because adenine is not having, so thymine is not having the same number of adenine molecules. If it was double-stranded, then adenine will be equal to thymine, guanine will be equal to cytosine. But the number of adenine molecules is different for number of thymine molecules. So it is a single-stranded DNA molecule. In red algae and in some caryophytes in green algae, the elongated apical portion of the female gamete, caprogonium or rugonium, that is receptive to male gamete is called a trichogyne. Answer is option A. Then next one. Which one of the following statement is not correct? Hybridization between different species usually result in sterile offspring due to failure of pairing at meiosis. That's correct. Cleistogamy is an outbreeding device. Cleistogamy. What is cleistogamy or cleistogamous flowers? In cleistogamous flowers, the reproductive parts are enclosed within the petals and they are not exposed. Hence, outbreeding is prevented in these flowers and self-pollination occurs within the permanently closed flower without any help of any pollinating agent. So, cleistogamy is an outbreeding device is not correct. Cleistogamy is an inbreeding device. They have asked for the not correct statement, so answer here is option B. Then next one. What would be the frequency of occurrence of the restriction site for PST1 in a DNA from thermophile with G plus C content of 80% in its genome? So CTG, CAG, just like how we did the previous one, we have to do here. But the only difference is they have told there is 80% in its GC content. So 80% GC content means 40% will be G, 40% will be C, 10% will be A and 10% will be T. Now probability that C will occur at position 1 is 0.4 or 40 by 100 because C is only 40%. Probability that T will occur at position 2 is 0.1 or 10 divided by 100. Then G will occur at position 3 is again 0.4. C will occur at position 4 is 0.4. A will occur at position 5 is 0.1. And occur at position 6 is 0.4. So multiply all these probabilities and you will get 0.000256. Now frequency of occurrence of the restriction site for PST1 with 80% GC content is 1 by 0.00256, that is 3906. Answer is option A. Now next one. Agent Orange used as a leaf defoliator by US Army during Vietnam War was. It was 2,4-D and 2,4,5-T, that is called as the Agent Orange. Then next one. Which one of the following brain centers in the human is called troubleshooter in visual function? It is superior colliculus. Answer is option D. White-tailed deers are most active during the twilight hours of dusk and dawn. Which one of the circadian rhythm describe them? Is it diurnal, diurnal, crepuscular, nocturnal? So they have told it is active during the twilight hours of dark and dawn. Now circadian rhythm, it is 24-hour cycle that are part of the body's internal clock. Diurnal rhythm, it is circadian rhythm which is synchronized to day and night. Crepuscular refers to twilight, the time short between sunrise and after sunset. And nocturnal, animals that sleep during the day and are active at night are called nocturnal. Here they have told they are more active during twilight hours of dusk and dawn. So it is circadian rhythm describing them is crepuscular. Answer is C because it refers to twilight, the time shortly before sunrise and after sunset. Now next one. Which one of the following cutaneous receptors is not activated by steady pressure stimulus? It is Meissner's corpuscle. Answer is option. Then next one. The following are some of the statements given for dosage compensation. In XX individuals, which one of the following options given below has all the correct statements for dosage compensation in mammals? So XX individual inactivation of one of the X chromosome occurs at early stages of development, not at one cell stage. That is correct. Then X chromosome inactivation is random, that is any of the two X's. Maternal or paternal gets inactivated. That's also correct. In female, SX1 mRNA gets alternatively spliced, thus producing functional SX1 protein. That is not correct because in female, SX1 mRNA are alternatively spliced due to the action of SX1 protein. So third is not correct. Then in males, hyperactivation of the only X chromosome compensates the dosage of X chromosomal gene. That happens in Drosophila, not in mammals. The question is for mammals. Then females are mosaics for functional maternal or paternal X chromosome. That is also correct. So the answer is D, that is 1, 2 and 5. Then next one. In humans, it is usually predicted that half of the babies born will be male and half will be female. Which of the following is the most appropriate explanation? Because of the random segregation of X chromosome during female meiosis. Because of the random segregation of X chromosome during meiosis. That is correct. That is male meiosis it should be. Because of the random segregation of X and Y chromosome during male meiosis. So sex is determined by the sex chromosome in the sperm that fertilizes the female egg. Half of the sperm have X chromosome and half have Y chromosome. So this is the reason for the question. So the answer is option B. Then next one. Membrane potential in intact mitochondria will favor finding of which one of the following compounds after passage through inner mitochondrial membrane, that is a cationic dye. Answer is option C. Then a series of 5 cell lines are created by human mouse somatic cell hybridization. Each cell line was examined for the presence of human chromosome and for the production of an enzyme. The following results are obtained. On the basis of the above result, the gene is mapped to chromosome number. So they have given the gene product here and the chromosome also. You have to match which chromosome is coding for this particular gene product. So A is produced, cell line A produces, so cell line A is producing in 1, 2, 3, 4, 5, 6, 7. It is B is not, cell line B is not producing. It is not producing in 1, 3, 5, 6. Okay. C is producing. C is producing in 1 and 2, then 4, 5, 6, 7. D is not producing. D is not producing in 3, 4 and 5. So E is producing. E is producing in 4, 5, 6, 7, 8, 9. Now if you see this pattern, that is plus, minus, plus, minus, plus. That is seen in chromosome 5 also. Plus, minus, plus, minus, plus. So the answer here is option B, that is 5. Sorry. At which of the following site the partial pressure of carbon dioxide is highest? That is systemic venous blood. Answer is option D. Which amino acid in the polypeptide undergoes end-linked glycosylation in the lumen through rough endoplasmic reticulum? It is asparagine. Answer is option C. Then next one. Which of the following marker system relieves higher polymorphism? So it is SNP, single nucleotide polymorphism. They reveal higher polymorphism. That is, the level of polymorphism is extremely high in SNPs. Next one. Drosophila melanogaster and Drosophila simulans are morphologically similar but are reproductively isolated. They are known as. What are sympatric species? They are most evolved from the ancestors that are existing in the same geographical area for a long period of time. There is no reproductive isolation observed in sympatric species. Allopatric species because of geographical separation between the two populations. And sibling species, they are morphologically similar but are reproductively isolated. So here they have told they are morphologically similar but reproductively isolated. So they are sibling species. Answer is option C. Then next one. Ex situ bioremediation technique involve excavation and subsequent transportation of pollutants to another site for treatment. Which of the following technique is not part of this process? So ex situ hybridization is the process of removing contaminated materials from its natural environment and treating it in a different location. So here, which of the following technique is not a part of this process, they have asked. So the answer here is bioventing. It is an in situ bioremediation technique, whereas all the other three are ex situ bioremediation techniques. Then next one. The two statements, one is assertion, another is assertion. The carbon dioxide content of the atmosphere has been rising in the past few decades due to burning of fossil fuels. Yes, that's correct. On an average, there is an increase in temperature of 0.4 to 0.5 degrees per decade. That is the reason for A. Now, because of this increase of carbon dioxide content, temperature is also increasing. So, both A and R are correct, and R is the correct result of A. Okay, not reason, R is the correct result of A. Then next one. Which one of the following characters of anthoceros is shared with that of green algae? Stomata like slime pods, intercalary meristem at the base of the sporophyte, amphithecial origin of archaeosporium, each cell with single chloroplast having pyrenoid. That is correct. The next one. In Escherichia coli, the mismatch repair system repairs the misincorporated base which is not removed by the proofreading activity of DNA polymerase. While doing so, the repair system has to decide which DNA strand is newly synthesized and which DNA strand is parental. Mismatch repair system does this by recognizing. So, mismatch repair system has to identify which is the parental strand and which is the newly synthesized strand. So, how does it recognize that? So, the daughter strand is unmethylated, whereas the parental strand is hemimethylated. This is how the mismatch repair system recognizes it. So, hemimethylated at GATC sequence nearby. Answer is option C. The deposition of pollen on the stigma of another flower is of the same plant is called as genesiogamy. Answer is option C. Then, which one of the following options have characteristic of non-poisonous snake? Presence of cylindrical tail? Yes. Broad transverse belly scales and the fourth infralabial scale largest? No. Cylindrical tail, broad transverse scales and third supralabial touches eye and nose. Presence of cylindrical tails and small belly scales. That is the characteristic of non-poisonous snake. So answer here is option C. Then next one. Juvenile hormone regulates development and reproduction in arthropods. Chemically it is a terpenoid. So juvenile hormone, chemically it is a terpenoid. Mutable collagenous tissue is the characteristic of the phylum Echinodermata. Answer is option D. Which of the following techniques would be the most suitable to identify a microdeletion? So if you want to identify a microdeletion, you have to go for fluorescence in situ hybridization technique. Fangs are specialized teeth used for venom deliverance in animals. In snakes, Solenogylophus fangs can be seen in So these types of ants, they are seen in vipers. Answer is option. Then baffles are metal strips attached radially to fermenter wall. The major function of the baffle is to break the vortex formed during the agitation process. Answer is option B. Which of the phytohormone is primarily involved in stomatal closure under drought conditions? So under drought condition it is ABA. Answer is option B. Which moiety of hemoglobin molecule binds to 2,3-bis phosphoglycerate at pH 7? It is the amino terminal. Answer is option A. The gram-negative motile bacteria species Pseudomonas carboxydovorans is an aerobic carbon monoxide oxidizing bacteria. Answer is option